to my ex-student Salman Sheikh, and see, he's the one who's just helping me to come live and doing all this thing. I'm very much thankful to Salman. Thank you so much. Now let us start our topic. So today basically we are here to discuss the semester 2 paper 1. Tomorrow is your practical exam. I know you all might be a little bit nervous, and you might be little bit like क्या पूछेंगे मैम, what is going to be that, but sure, everything is just only from your syllabus. Okay, so let us move ahead without wasting of time. Actually, what has happened, I don't know why I am not able to hear my live stream in my mobile, and that's what is just jumbling me whether am I audible or not. And I was just troubling Salman, help me, and that boy is also helping me throughout. So now I'm sure my children are telling I am audible. So yes, thank you so much for confirming it. Once again, I will ensure in my another device that I am audible. Okay. So when we talk about the semester 2 paper 1, so basic, now see in my another device also my voice is not audible to me. I don't know what is the problem, but I'm audible to you. That is just sufficient for me, and that is more than silent. My voice is not audible to me. I don't know what is the problem. Okay, I got it. I could hear mine. Okay, chalo. So let us start with this. So your paper is for the 2 hours. Now see student, this 2 hour is really very crucial for you all for writing your examination, and the paper is for the 50 marks. So how this 50 marks is distributed? First question is your called as your major question. So this is going to be a performing question, and in that there will be 2 performing practical, but it does not mean that either this will come or that will come. You always know that we have the system in which we give the X and Y type, right? So the X student may get one of it and Y student will get another one. So one question number 1 first is estimate hardness from given water sample and compare the result. So you will be getting the water samples, and in that you have to find out the hardness. Calculation part is also there, or you will be getting estimate the free carbon dioxide from the given samples and compare the result. So here you will be getting a tap water or maybe the distilled water, and comparatively you will be getting the aerated water. Quickly we will just see the procedure how we all are going to have this. So first we will be talking about the estimation of hardness of water. So we will be taking 20 ml of the standard water sample. Now student, here the procedure will be absolutely same for all the waters whichever you are getting in, but only the thing is that you will be applying the same procedure. This protocol will be given as per the university rule. So first you will be taking the 20 ml of the standard water sample. What do you mean by the standard? Just a minute. What do you mean by the standard water sample? Here means the whatever sample you got it. Okay, and after that you will be adding the 2 ml of buffer solution. Now see student, we all have learned already that this buffer help us because this EBT works under the buffer situation only. So we will be taking the pH 10 buffer, and after that you will be adding 2 to 3 drops of the EBT indicator. So first step, you have added the standard sample water. After that you have added the 2 ml of the buffer solution. After that we will be adding the 2 to 3 drops of the EBT indicator. After that, first you will be rinsing the burette with the EDTA solution, then fill it. Now see why there is a need to rinse the burette, so that the pH will maintain. So this is a very crucial step. Usually what happens, student directly fill the burette and after that start performing. No, the ideal way is that you have to rinse the burette. That means you fill the burette, then empty it, then again fill it with the EDTA, and after that you will be titrating it. Once you have titrated, note down the reading into the table, and you have to repeat the titration to get the concurrent values. Now how many titrations you have to take, that will tell you into the day of the examination. So steps are simple. First you have taken the conical flask, in that the, if two samples, three samples, whatever samples and whatever titrations are there, that will be told. After that you will be adding the 2 ml of the buffer solution, you will be adding 2-3 drops of EBT indicator, and you will be rinsing the burette with EDTA. Then after that you will be titrating it, noting down the burette reading, repeat the titration to get the concurrent value. We'll take a small pause for the azan. Okay, so thank you so much, Salman, for your overall support. Definitely, when the students helping you in a way that you could do something in the life is really very great feeling for a teacher, and that's why I'm very much thankful to Salman. Also, come back to the point. See, we were discussing the question number one. In the question number one, I said there will be two practical. Student, I could not see after a tiring schedule. If I'm coming online for you, it is expected all of you must also join me. So it is really bad that only till now 5 students have joined, others haven't joined, although I have declared the schedule into the college only. So this is really not an appropriate attitude. Okay, so question number one, I'm not going to post this live stream, because this is beneficial only for those who have joined me right now. I'm not going to share it, and this will not be available after the live streaming. Chalo, let us discuss the question number one. As before Adan, we all have discussed the question number one is the two question will be there. First question, estimate hardness from the given water sample and compare the result. So this particular question is for 15 marks, and this is the only performing question you all will be having it. Now see, in the question number one, first option we have already seen, it is the hardness of the water, and hardness of the water, the protocol I have already explained you. Now we will see the second, that is the comparing the result of the carbon dioxide. So question number one, first option, hardness of water, you all will be taking the 20 ml of the standard sample of the water, In that you will be adding the 2 ml of buffer solution, 2-3 drops of the EBT. Then after that you will be rinsing the EDTA and rinsing the burette with EDTA, and after that you will fill it. After that you will be noting down the burette reading, and this burette reading you will be comparing it and utilizing for your calculation part. I'm not going to discuss the calculation part right now, that is already been explained to you. But yes, if I will get the handbook in my laptop, then I will explain you the calculation part also. You will be repeating the titration. Let's see if I have the handbook in my laptop, then I will explain you the calculation part as well. Yes, I do have a handbook, so I will explain you the calculation part as well. This is the only. So see how we will be doing the calculation. Hardness in terms of the calcium ion, and here you will be taking the mean burette reading. Now see, you will be doing it separately. Like first you will be finding the mean burette reading for the sample 1, then sample 2, sample 3, whatever sample has been provided in the examination. Ideally there will be 3 water samples. You have uh, just sample A, you have to write sample A, B, C, whatever is given in your examination, in your exam timing. So that way you will be writing the hardness in terms of calcium ion for sample A, for sample B, as we have done into the regular practical. So if you will be asked to take the 3 readings, so you will be writing the mean burette reading that time. So mean burette reading into normality. Now see, here you will be writing the normality of the EDTA into 14.08, because 1 milli equimolar of EDTA is equals to 40.08 mg of the calcium ion. Then after that you will be multiplying it 1000, because we will be taking the final reading in a liter, and after that you will be dividing it with the 20, that is the 20 ml is your volume of sample. So this is your calculation part. Now let us solve the this second point. One point to be noted, that you will be getting wine red to blue color as your end point. This is your observation table, which I have given you in your regular practical, that only you will be solving it. This conclusion is really very important for the Viva point of view. Tap water was found to be less or more hard as compared to well water. Permissible value for hardness is recommended by WHO in 1993 is 100 mg per L, that is the liter. That's why we have multiplied it with the 1000. Both the samples are within or out of the range of the WHO standards that you have to mention. If your answer is less than 100, then or more than 100, that you have to mention. Okay, so because WHO says that it should be less than 100 mg per liter. Now let's move on the next practical, that is estimation and comparison of the free carbon dioxide. Now this reaction is important for this particular practical. Now see, in water, whatever carbon dioxide is there, that combines to give a carbonic acid, and carbonic acid when mixed with the NaOH, so it forms the sodium bicarbonate. We all know acid plus base is equals to salt plus water, and this is the general reaction we know. So this important reaction you must know, and this sodium bicarbonate, when it has been formed, so that presence is can be identified by the phenolphthalein indicator, because phenolphthalein indicator turn pinks when the pH is 8.3. So let us see the procedure for this. So the protocol for this particular practical is, we will be taking the 100 ml of the water sample, and after that we are going to filling the burette with N by 44 NaOH solution. You will add 2 drops of phenolphthalein indicator, and after that we will be titrating it against the N by 44 NaOH solution. Now end point you need to remember is colorless to pink. So as soon as a first pink image will develop, what will happen when you will be adding the drop of it, so pink color will appear. That after that it is going to disappear, so you add 1 or 2 more drops in that. So when you feel like that the pink color is stable, see student, do not keep on adding NaOH, add it drop by drop. So the first pink color when it is appeared, after that only 2 and 3 drops adding is advisable, and after that you will be taking 3 readings or whatever readings you will ask to take, that you have to take, and after that you have to calculate the free carbon dioxide and you have to compare the result. So you all know how to make this observation table. I'm not going to explain, I'm just going to explain how you all are going to take the calculation part. So free carbon dioxide of the sample, the formula is MBR into 44 into an into 1000 divided by V, that is the volume. MBR now you all know, N by 44 is, that's why we are multiplying it with 44, then you will be taking the normality and 1000. So this is all about the performing practical. Let us move on the next question. Student, let me know if you feel like I am going very fast or you have any doubt. Madhiha is asking me, ma'am, do we have to learn the protocol? No, Madhiha, protocol will be provided. Only you must know the procedure of performing. Alright? What you are going to write for practical question number 1, protocol we are giving. So you will be writing the aim, you will write the requirement, you will be writing the, if any chemical reactions are there, you will be writing the observation part and observation table as well as, and you will be writing the calculation. I repeat once again, you will be writing the aim, requirement, principle, and after that you will be drawing your observation table. And as I told you, in the observation you will be writing what is the chemical into the conical flask, chemical in the burette, as we have done in our regular practical, then end point preferably. Then after that you will be writing your observation table, and at the end the most important is the calculation. Alright, and protocol will be provided. You already have given your semester 1 exam, you know. Now let us discuss the question number 2. So when I talk about the question number 2, question number 2 is not the performing type. Here you will be solving the problem. So in the question number 2, you will be given 2 problems. Each problem have the 5 marks weightage. So any 2 is mentioned, it does not mean you will be having the 10 problem and out of 10 you will be solving any 2. Sorry. Here whatever 2 question will be displayed on the screen, both the question ideally you must know. If you don't know, you will not get the marks, and each question will have the 5 marks weightage. Now question will be asked from the natality, mortality, sex ratio, fecundity and the population density. So whatever questions we have solved in our regular practical, you all must solve it and you do the practice of it. So any two question will come. Writing formula ideally have 1 marks, and after that the calculation part. Then after that you all will be having this calculation part and interpretation. यानी कि writing the answer into the words you will be getting the marks. So overall formula पे 1 marks. Then after that solving you will be having 3 marks, and interpreting the result will have the 1 marks. Okay, for both the problems. Next is identify the brand animals. Question number 3 is really very important. Okay, question number 3 में you have to by heart overall the part which we have done into the national park wala this unit. So question number 3 में first option is identify brand animal minimum 4 and place them in their respective national park sanctuary on the given map, quoting the reason for their declining. Now see how you will be solving it. Here 4 animals or maybe the 5 animals will be displayed on the screen. Okay, out of that you have to just identify the name of that animal, and after that you must be aware that this particular national park this particular animal belongs to which particular national park, and after that you will be marking it into the map. So how you will be solving it, I will explain you with the help of your textbook only. I am thankful to Uber because immediately he helped me to take this textbook. He made immediate PDF for me. So thank you so much Uber for this. Now see here. Say example on your screen. Royal Bengal tiger or the gharial is been displayed. Now what is our question, student? Identify the brand animal. Okay, we have to write down their name, and after that you have to mark. So here say example Royal Bengal tiger is on your screen. So you have to identify Royal Bengal tiger. Now what is your question? Here only let us come on the question paper. This is your question paper, right? So mark the national park and sanctuary. No, sorry, this one. Identify brand animal. So brand animal is shown to you. You have to identify that brand animal, and after them place them in their respective. So identify means you have to write down that this is the Royal Bengal tiger. Place them in their respective national park. So you have to write on your paper that the A is Royal Bengal tiger belongs to, or it is the representative species of Jim Corbett National Park, and after that you will be provided the India map. So like this you will be mentioning it or marking it in the map of India. Alright, any doubt in this? Shall we go ahead? So what things you must be aware of? You must be aware of the animal on the screen is Royal Bengal tiger. This is an endangered animal. This belongs to the Jim Corbett National Park, and the most important thing is Jim Corbett National Park belongs to the Uttarakhand, and you should know the marking into the map. Okay, so it is the north marking. This marking is really very important. And the second animals is the gharial, which also belongs to the Jim Corbett. Now see, in the last practical I had given you this name of all the animals, their scientific name. Even we have discussed this particular animal belongs to which particular national park and their marking. This part we have done, and also we have written their status. Now their status is important for the next practical. Now second animal is the gharial, that is the critically endangered species. Then the next, Sanjay Gandhi National Park. Let us see the representative species of the Sanjay Gandhi National Park. Sanjay Gandhi National Park, it is in Mumbai, Borivali, Maharashtra. So you remember its marking on the map. It is present on the western coast. Okay, तो यहाँ पर the representative species of the Sanjay Gandhi National Park is the leopard. Leopard is vulnerable species. Rusty spotted cat. Rusty spotted cat is near threatened species. Anti means near threatened species. Let us go fast to the Tadoba National Park, which is which is also called as the 43rd reserve under the Project Tiger. Now it is also in Maharashtra. Why it's not moving? Maharashtra northeast में है ये. Now representative. See Maharashtra. This is the Maharashtra state northeast. This side. Okay, this is really important to remember the marking into the map, student. Now representative species is the Indian pangolin, which is endangered, and the next is forest owlet, which is the critically endangered. Let us go to the Kaziranga National Park of Assam. See, this is our Assam ka Kaziranga National Park. Ganges dolphin and one-horned rhino. When I talk about the Ganges dolphin, it is endangered species, and when I say the one-horned rhino, it is vulnerable. You need to remember the marking into the map. It is in Assam. So what we will remember about one-horned rhino, Ganges dolphin, then its marking on the map. Next is the Gir National Park, which is also a wildlife sanctuary. So this is present into the Gujarat. Let us see the representative species of the Gir National Park. So Gir National Park Gujarat mein hai. You need to remember its marking into the map. So it has two representative species. The first is the Asiatic lion and the long-billed vulture. Asiatic lion, it is endangered, and this long-billed vulture, it is critically endangered. Next national park, Silent Valley National Park. It has two representative species, lion-tailed macaw. It's in Kerala, and Malabar spotted civet. That is the critically endangered. Pyrothon Island Marine Park. गुजरात में है ये. Now coral reefs. See, now if I will talk about Gir, it was somewhat here, and this is somewhat into the west sides. And the next is the dugong, which is vulnerable. Let us move fast to the next national park, that is the Koladev Ghana National Park, which is present into the Rajasthan. Marking into the map this side, okay, northeast side of the Rajasthan state. So the representative species is blackbird, which is near threatened, and Siberian crane, which is critically endangered. Next is your Bandipur National Park, and it is also a wildlife sanctuary. It is located into the Karnataka. For quick revision, I had already given it in your journal, right? So you should definitely go through with the journal. Those who are absent, make sure that you will be watching it and you are going to do it. I am not going to save this live stream because I had already told you all to join online. So those who have not joined, they have missed it. Sorry. Then next is the Asian elephant. So Asian elephant is endangered, and red slender loris, it is the endangered species. So it is in Karnataka, Kuldev, this is the wildlife sanctuary Kuldev. और ये जो है यहाँ के दो representative species is the Asian elephant and the red slender loris. So this is all about our question number 3. Let us move back. So question number 3. Question number 3. Question number first option, identify brand animal. For this you will be having the animal on the screen. You have to identify the name of the animal. You must know this animal is the brand animal of which particular national park. You must know that national park is in which state. You must know its correct marking on the map. Alright, let's move ahead and move fast to the next option. Mark national park and sanctuaries on the map of India and mention the name of their brand animal, stating reason for their decline. Now here you will be given names of national park. Any 5 national park will be given, or maybe directly 4 they may give. If 5 or 6 will be given, you will be asked mark any 4 national park into the given map of India, and after that on your answer book you have to write this national park belongs to which particular state and which are the brand animal. Alright, so here in this option you will be given the names of the national park and you will be asked to identify, you will be asked to mark it on the map and you will be asked to write down their brand animals. In your theory, it is a reason for the decline is mentioned. That also you have to write. Now question number 3, you will be given any animal. It may be the endangered or it may be the critically endangered. You have to identify that particular animal, the status of that animal. Now here what you are going to write, we are expecting that the student must know the name of that animal along with the scientific name of that animal. Okay, you must know the scientific name of that animal and you must know the status, whether the animal is endangered or the critically endangered, and after that you have to write the reason for their decline. Alright, let us move on the question number 4. If anyone have any doubt in question number 3, please tag me right now. And if you don't have any doubt in question number 3, so please tell we don't have any doubt so that I can move ahead. So does anyone have any doubt in question number 3 or everything is clear to you? Are you with me? Okay, I think no doubt is there. So let us go ahead. Don't have any doubts. Okay, Sumiran, thank you for replying. Okay, so let us move ahead on the question number 4. So question number 4, the first option, see अब question number 4 में lot many option is there और इसमें से कुछ भी आ सकता है students. Study the given information and give answer on the basis of the food chain, food web and the ecological pyramid. So here either you will be having the food chain or food web or the ecological pyramid. Any one thing will be there, and on the basis of that you will be having a question. I will give you a simple example. I think I may have it in my this ecological pyramid only. I will show you. Say example, this this particular ecological pyramid is given to you. Okay, this particular ecological pyramid is pyramid of number. So say example one ecological pyramid, any ecological pyramid is given to you. So here there will be very simple questions that identify the type of ecological pyramid. Say example this particular pyramid, this only pyramid will be shown to you and here numbers are there. So we should know that this particular which type of pyramid is this, which type of ecological pyramid is this. So this is the ecological pyramid of number because here the number of organism on each trophical level is mentioned. So simple questions will be there, which trophic level maximum animals are there, minimum animals are there, like these questions will be there. So as I have opened this PowerPoint, let us quickly revise the ecological pyramids. So ecological pyramids we have seen are of three types. One is the pyramid of number, in which the animals this particular pyramid is made on the based of the numbers of organism on each trophical level. So here on the food chain, always the primary producers are more, then primary consumers are less, secondary consumer will be less, and tertiary will be the least into the population. Why? Because only 10% energy passes from one pyramid to the second pyramid. This is your laboratory exercise. Now construct an ecological pyramid of number with the following data. This we have done into the regular practical. Now pyramid of biomass, you need to remember that biomass, that is the dry weight of organism at each trophical level at one time, is mentioned here. Okay. Now next is this is the regular practical we have done. Ecological pyramid of energy based on the amount of energy passing to each trophic level. So we have learned generally the 10% energy is only passing from one trophic level to the next. Then next we have, this is the old one, paper 1 it is. I am not going to explain you this. Okay, alright. So such type of questions will be there. There may be the question in which the J curve or any other curve will be there. Food chain will be there. So there will be simple questions on the basis of the food chain will be asked. There may be the food web will be there. So the question on the food web will be there. So you will be asked the question, identify the food chain. So you need to understand that this particular food chain is of grassland or maybe the desert by knowing the animal, because we have done lot many food chain and food web into the regular practical. Now question number 4. Prepare the food chain, food web and the ecological pyramid from the given data and give its significance. Now see here student, prepare the food chain and food web. Sometime it may happen, as we have done into the laboratory exercise, we were given the names of some animals and we were asked to prepare the food chain, right? Do you remember the animals, scattered animals will be there? I will show you one regular practical exercise, so that will be more and more clear to you all. See this time, such food chains will be given, or you may be directly ask a question, draw the food chain for the grassland, draw the food chain for the fresh water, draw the food chain for the marine water. So such type of question could be asked. See, this is the food web. So a food web will be given, on the basis of that animals will be there. Like this, aquatic food web, construct the food web using the following animal recorded in the ocean. So these animals will be mentioned and you will be asked to draw the food web on the given detail. Alright. So such type of desert food web, any type of food web could be asked. Alright. And there was one little activity to given to you all. You were given an activity that you have visited this Maharashtra Nature Park, right? In that you were asked to draw the food web of that. I hope you all have done that particular thing. So this is the second question. Let's go on the next question. Identify and interpret the given graph, growth curve, age structure, and comment on the pattern of the population dispersal. So here, growth pattern like J-shaped curve, S-shaped curve will be given, sigmoid curve, and the J-shaped curve will be given to you. You will be asked, or maybe the population dispersal, age structure. We have done Ethiopia and all that. You remember? So that type of questions will be given, and the questions will be asked to you. I will just go through quickly on this. On the question number 4, this option. Because this we have done into the beginning only. Yeah, this. So this type of graph will be given. On the basis of that, you will be asked to write down the question. So interpretation of the given graph, tables, and comment of the pattern of the population in the nature. So three type of curve we have seen. Highly convex curve that belongs to the type 1 species, those who live their maximum lifespan. Diagonal is actually depend, these are the predator species. Their life certainty belongs on the animals, those who are predator on them. So prey species usually आएंगे. Highly concave curve is having the highest mortality. तो यहाँ पर जो है you will observe that they are having the large number of the eggs like fish, oyster and all. Life. So basically this type of pyramids will be given. This I wanted to tell you. See, pyramid of age structure, then bell-shaped age structure. So age structure are of three types. Pyramid age structure. This is for the you can say the developing countries. Then bell-shaped structure. It is for in which the developed country we can say where the number of reproductive and reproductive same. Unshaped. So these are the three we have learned. So such type of, basically what I wanted to say is such type of graph will be given to you. Now then determine the population density by the capture and recapture formula. That we have done. Sanjay Gandhi National Park population was given, and after that we were asked to do the capture and recapture. I hope you all remember that. So all such questions we have already solved. I am not going to repeat the solving part now. Now the last question is the journal, certified journal. I hope everyone have certified their journal. Do you have any doubt in question number 4? If yes, tell me where you have a doubt, because now I will end this stream. So if anyone have doubt, let me know, because this is the last question. After that, I am going to end the stream. Okay, if you don't have any doubt, we can go ahead. Journal and the FYBSC. So this particular question, your certified journal, the date of certification have a real meaning. So make sure that before entering, thank you Sumiran for confirmation. Make sure before entering into the lab, you have a certified journal in your hand and your journal is properly covered, and you will be asked some viva. Okay, question number 4. Hasina, just now I explained question number 4. That question number 4, first option you will be given. Just now I have explained it. That question number 4, first option you will be given the food chain or the food web. In that you will be asked some questions. तो वहाँ पर ready-made diagram will be there or the graph will be there, and on the basis of that question will be asked. Okay. So, and in the second option, you will be having a question, prepare the food chain or the food web. You will be given animal or directly you will be asked to prepare the desert food chain or the food web, grassland food chain, food web, whatever. So, either you will be asked to prepare the food chain by the given data, and see here the most important thing, writing significance have marks, because this particular question is for the 10 marks. So, you have to write the significance of it, and the significance is mentioned into the background information if you will read carefully. Then the next third option, you have to identify and interpret the graph, growth curve, age structure, and after that you have to comment on the pattern of the population dispersal. The last option in that is the capture recapture method that we have done in our regular practical. It is a calculation part. So if the capture recapture type of question is asked, you will be having the two questions and you have to solve both the questions. Okay, now last, I hope Hasina your doubt is clear. If it is clear, please confirm it into the chat box, so I will also be free. Yes, it is confirmed to you. Yes, Hasina, is it clear to you now? Okay, bacha. Now let's move ahead on the question number 5 quickly. Everyone must have their certified journal, and you will be asked a viva voce, that is oral questions will be there on the basis of the practical component. Okay, so here before entering into the lab coat, all must wear a entering into the lab, all must wear a very clean, neat lab coat. Everyone must have the equipments or apparatus asked to bring by your examiner. For paper 2, you have not got such instruction, but only thing which you need to carry is your calculator. Make sure you have the one or two calculation questions will be there. So everyone must carry the calculator, and your all stationery, all right, whatever you required, and whatever paper 2 teacher has instructed to you for carrying, you all must carry that. So those who have joined late, see quickly fast, I'm going to revise the complete paper pattern. Semester 2 paper 1 is 2 hours 50 marks paper. Okay, in that question number 1, estimate hardness from the given water sample. Here you will be given the various water sample like tap water, well water, pond water, distilled water, or whatever type of water, whatever is your examiner has decided. And after that, estimate the free carbon dioxide from the given samples. So here there may be the sample of the aerated drink, tap water, distilled water, different type of samples, but here we will not tell you which type of samples are given. For both the options, samples will be marked as A, B, C, D. You have to identify that this particular water is of what type by your regular practical reading. Okay, so quickly we will see the procedure once again. For question number 1, estimation of hardness, that is your first option. You have to take the 20 ml of the standard sample water. In that you will be adding the 2 ml of the buffer solution. After that you will be adding 2 to 3 drops of EBT indicator. In that you will rinse it, rinse your burette with the EDTA, and after that you will fill it, titrate it, and note down the burette reading into the table. Repeat the titration to get the concurrent values, and after that you will be doing your calculation part. The second option in question number 1 is estimation of the free carbon dioxide from 2 different samples, aerated drink and the tap water. So in that you will be taking the 100 ml of tap water and 100 ml of the aerated drink, and both the flask you will be marking as A or B. After that, fill the burette with N by 44 NaOH. Now see, for marking the flask and all, you must have your permanent marker with you. Okay, permanent marker will not be provided. So permanent marker, calculator, everything you must carry your own. In exam, if you will be asking it to your friend, definitely we are going to make a copy case against you. We are not, at any moment we are not allowing you to talk, even for asking the stationery. It is not allowed, and strictly not allowed means not allowed. So make sure you all will carry your calculator with you, and don't tell we were not aware. You are not aware, that means you are not regular into the paper 1 practicals. The student, those who are regular for the paper 1 practical, they know very well that for paper 1 practical they definitely require the calculator. All right. So here you will be filling the burette with the N by 44 NaOH. Say the rule follows here also. First you will be rinsing your burette with N by 44 NaOH, and after that you will be filling it. For indicator, you will be using the phenolphthalein indicator in both the flasks. Titrate against N by 44 NaOH, and you will be titrating it till the pink color observed, because the once your solution is turning into the pH 8.3, definitely you will be getting a pink color. Then question number 2, solve the problem. Now this problem will be based on the questions we have done: mortality, natality, sex ratio, fecundity, population density. So all this question will be asked. Then question number 3, the first option, you will be given the animal on the screen. You have to identify the brand animal. I'm sorry. Identify that particular brand animal. Write down that animal name in your answer book. I'm sorry. You have to identify that particular animal, and here the you will be writing that animal's national park name, as well as you will write the name of the state in which that national park situated in, and after that you will be marking it into the India map which is provided to you. Second option, mark the national park. Here on screen, name of the national park will be displayed, and it is expected that you should know. Now earlier I have already gone through with the revision part, which animals belongs to which national park or the sanctuary. So here name of national park will be mentioned. On the answer book you will be writing their brand animal name, as well as you will be marking it on the sanctuary, and you will write that why these animals are declining. All right. Now third option, identify endangered and critically endangered animals. So here photographs will be displayed on the screen, and after that you have to write the reason of declining that particular national park. Question number 4. Now student, question number 4, there will be 4 options. Study the given information, give answer on the basis of food chain, food web and ecological pyramid. So out of these 3, any one will be there on the screen, either the food chain, food web or the ecological pyramid. On the basis of that, question will be asked, and or second option, you will be given the name of animals and you will be asked to prepare the food chain or the food web, or directly you will be asked prepare the food chain for the grassland, for the desert or aquatic animal or whatever, food chain or food web. And after that, next option, see here only in second option, once you are done with the food chain and the food web or whatever ecological pyramid question which we have solved into the regular exercise, in that you will be asked to on the basis of that you will be asked to prepare the ecological pyramid. Now here you have to write its significance. What is the significance? The significance is mentioned into your background information, and that writing significance have 2 marks. So students, it is really very important that you will be writing the significance. Now next option, the option 3 is identify and interpret given graph, growth curve, age structure. So given graph, we have just seen that there are type 1, type 2, type 3 animals. Various graph are there, concave, convex, and the diagonal. That will be their growth curve, J shape, S shape, or the age structure. Age structure is also of 3 type: pyramid age structure, bell shape, on shape. So this type of structure will be given, and you will be asked to comment on the pattern of the population dispersal, which we have done into the regular practical. The last question: determine the population density by capture and recapture method. So that we have solved this is the calculation type of a question. We have solved it, that the number of animals are given and they are been marked, and after that they were released into the wild, and after that they recaptured. So you have to find out it with that particular method. And the last question, journal and the viva voce. So in that, it is expected that the student must have certified journal with them, and they will be asked the viva on the basis of the practical component. So I wish you all the best for your exam. If you have any doubt, let me know. And at the end, definitely I like to thank to Salman, the FYBSC student, who is the pass out student from the 22 batch. I'm really thankful to him because he's a YouTube blogger, and he helped me to join this YouTube stream through the OBS studio. And also thank you so much, Salman, for being with me. He is very busy, but out of his busy schedule he helped me because I wanted we have to uh, go through with this practical part. Now Kahkasha Beg is asking me, ma'am, for the first practical, do we have to learn requirement? Yes, my child. You must know aim, requirement, and principle. I repeat, you must know aim, requirement, and the principle for your practical number 1. So if anyone have cleared with their paper, so I wish you all the best, and I will end this stream. All the best for your examination. Do you have any doubt, or should I end this stream? So I don't think you have left with any doubt. So I will just end the stream, and I wish you all the best. Bye everyone. Thank you so much. Thank you so much for joining. You all can leave now. Bye everyone. Thank you so much for being with me. You're welcome, Hasina. Bye beta. Good night everyone.